This presentation, Testing Biodiversity, Microcosms as Predictors of Ecosystem Capabilities, presents the objectives and findings in a microcosm-based experiment. This microcosm was intended to build understanding of how balance in an ecosystem determines its fate. When I started this experiment, I was hoping to learn which trophic level has the most impact. Microcosms can be useful tools in ecology, providing a convenient model of how changes in an ecosystem may affect its overall health. It's important to understand that the species represented in this experiment were selected at random and remain mostly unidentified. My hypothesis is if the contents of the microcosm are an accurate representation of the ecosystem from which they were acquired, then life at all trophic levels will persist for the duration of the experiment. If an improper ratio of autotrophic life is present in the system, then heterotrophic life cannot persist. Materials for the experiment were collected at Pikeview Reservoir, a man-made reservoir for the containment of water to be treated for municipal waste. This is an open-air reservoir stocked with fish, frequented by ducks and other waterfowl, and adjacent to a major city street. It does tend to contain trash, stormwater runoff. Specifically, the points indicated on the map and in the respective picture, which is a zoomed-in version, will indicate where the water and species were collected. The system was constructed in a 500 milliliter plastic water bottle. Approximately 254.5 cubic centimeters of reservoir bottom soil were added to the bottle. Additionally, 184 cubic centimeters of water from the reservoir were then placed into the bottle. 4 stalks of aquatic grass found in the water at the collection site with their roots were then added. A small sampling of algae was then placed into the bottle. The exact amount is difficult to quantify. I would estimate that to be approximately a teaspoon of algae placed into the system. When it was returned to my home, I was able to identify 2 aquatic worms. These are of unknown species. Further research did indicate that these are likely tubifex worms, but I cannot verify that. Also, I was able to identify in excess of 10 small red larval stage species. Research here would indicate these to be of Chironomidae. That would be a midge, a non-biting flying insect, very common in some of these stagnant water areas. Easily mistaken for mosquitoes. I have had pretty significant experience with these, and I'm confident that that is in fact what they were. Other things I was able to control in this experiment would be the lighting. The room in which the microcosm was held does not receive really any natural light. Light was provided by an artificial source, that being a lamp at my desk. The lamp is equipped with Sylvania 8.5 watt, 800 lumens LED daylight bulb, as close to full spectrum as I was able to provide. I'm sure that the ultraviolet capabilities of this light are limited. However, it did afford me the ability to control the photo period, which was maintained at approximately 15 hours a day, which was based on the idea that at the time the sample was collected, that would have been the average photo period in the natural environment. Temperature in this location was regulated by my home thermostat at approximately 20 degrees Celsius. Again, very comparable to the environment in which the species would have naturally been occurring at the time of collection. The experiment lasted for a total of 6 weeks, beginning Sunday, the 13th of June, and concluding will actually be future of this recording, Sunday, the 25th of July, which will be tomorrow. On day 1, as we discussed, the heterotrophic life was abundant. I was able to identify the 2 worms, again, believed to be tubifex. They are identified readily in the photo. We can also see here several examples of the midge larva. One here that is blurred, and a portion of one is visible at this location. On day 1, the water was fairly opaque and had a pale green color. Little organic matter was visible in the soil. The grasses that I had added to the system, again seen in the picture on the previous slide, really represented the vast majority of organic matter. We are attributing the pale green color to the algae, as this was not readily identified in large clusters. Soil had a rough texture, moderate to high gravel content, however it was primarily composed of sand. Within days, the water had clarified completely. It was crystal clear. Species seemed to be very active. The 2 large worms had a tendency to adhere to the sides of the bottle. Occasionally they would actually climb to the airspace above, and they were very apt to reach out. Initially, I took this as a predatory behavior. However, additional research leading me to believe that they were tube effects indicates that they were probably more likely collecting particles of decaying plant matter from the water. By the conclusion of the first week, however, activity in the system had slowed. The algae did settle to the bottom. It was very observable at that point. The water did remain clear. Here we see the system halfway into the second week. At this stage, all heterotrophic life had ceased. No movement, no evidence whatsoever, and we can also see that the aquatic grasses had died and floated to the top of the system. The pale green color is starting to return, which indicates that there is some algal growth happening, but no other life to be seen. Less than a week from that photograph, we do see that the water took on a very turbid darker green appearance. This photo was about a 3 to 4 day gap from the previous. The growth was so explosive that I was not able to capture an interim photo. At the time of this writing and the termination of the observation period, the microcosm remains in a very similar state. This is the current status. We do see that the grasses are still floating at the surface, but there is no additional growth in those life forms. This experiment allowed me to determine that the heterotroph suffered from a lack of some resource. Likely, we are looking at a food deficiency, given the limited amount of autotrophic life that was initially added to the system. Different species of midges do feed on different sized particles, but are generally detritivores and or feed on algae. The small amount of algae initially present, combined with the small amount of plant matter to die and decay in a sufficient amount of time, likely led to the downfall of the autotrophs in this system. Again, if the worms were in fact tube effects worms, they also would have been detritivores. The 2 species that we're discussing here, the midges and the tube effects, would have been in direct competition, most likely outconsumed the resource production rate of this system. The apparent collapse followed by the sudden explosion of the algal population can most likely be attributed to the loss of the limiting factor of the consumers, with nothing there in the form of those midge larvae to consume the algae as it grew. There was nothing to stop it. The abundance of light, the nutrients provided by the waste left by the autotrophs while they were present, and then of course the decay of their bodies would have provided ample nitrogen. There's no lack of light for photosynthetic activity, and we can assume that the airspace provided a relatively high concentration of CO2. These findings do support the hypothesis that an inadequate autotroph to heterotroph ratio will not allow for the persistence of a diverse community. Consumers were limited by the availability of resources to the point that they were unable to complete their life cycles. No reproduction was noted. The larval midges never were able to emerge into adults. This is also probably attributable to the fact that there was a limit of abiotic materials, that being there were no large rocks protruding from the surface of the water where these larvae could adhere and complete the life cycle, emerge into adults. Even if that were a possibility for them, it is unlikely that those adults would have survived to reproduce given the lack of resources. Course concepts that are clearly evident in this experiment are the issues facing ecological systems. The experiment demonstrated that a lack of biodiversity poses a serious threat to success of ecosystems. If there was a predator present in the system, it is possible that the algae may have grown faster before the loss of the heterotrophic life. Therefore, it may have been able to support a continuation of those consumers. Perhaps we still would have lost the midges given the lack of abiotic factors, but it's very possible that the tube-effects worms would have continued to grow and multiply as plants were dying in the system and providing them detritus to feed on. The next concept is the importance of conservation biology and the anthropogenic impacts to the landscape. Effectively, my selection of a limited density of autotrophs was the downfall of this system. More food, greater ability to cycle nutrients would likely have provided greater success. Again, we're looking at the midges probably would not have made it without some of those other factors. But this really is the essence of the discipline of conservation biology. Looking at human impacts, what we do to these ecosystems in adding, removing, limiting species and or resources can take a huge toll. The final concept is the analysis of data in terms of real-world application. The experiment shows in a very short time how that imbalance in an ecosystem can result in the total dominance of a single species, which may ultimately itself fail due to a lack of contribution of resources from other species in the system. This plays directly into my hypothesis for the future of this microcosm. If there are no other life forms to contribute to nutrient cycling, that being in the form of excrement providing ammonium nitrate to be fixed into usable nitrogen, then the algae-dominated environment will not be able to persist. In preparing a similar experiment in the future, I would spend more time examining population densities in the natural source and work to more closely emulate those to test the ability of the system to persist without further anthropogenic interference. Again, when these system components were selected, there was little time dedicated to examining water depth versus the surface area from which the sample was being taken, the relative abundance of species. The midges were not observable in the wild. It was not until the microcosm was brought home that I was even able to see those types of things I would take more time in examining. The use of microcosms as a tool to study ecosystems is relevant to the field of general ecology, as it provides a means to test how changes in particular factors can affect overall ecosystem health, from addition or removal of abiotic factors, the rocks that we discussed, decrease in water, will a lower abundance of water stress a plant population to the point that it becomes extirpated, that predator addition. All of these questions can be addressed without having to worry about impacting the ecosystem in its natural environment. These sources were referenced in the compilation of this presentation.